let's introduce the model cell in which, as I said, every reaction will be able to occur. As a little context on this figure, I have decided to create this model because, while learning the metabolism of macronutrients myself, I wish that there was a resource that would integrate all the reactions together to have a good visual summary of what's going on. As such, I have been working for the past few months on this map that I hope will be useful for others to study metabolism. With respect to what we will cover in this video, there is much more information on the map. That extra information will be useful in future videos to cover other nutrients, but for the moment, we will only consider fats and carbohydrates. If you want to visualize this map on your own, there will be a link in the description to download the map and the associated legend. Now, you can see that the map is complex and has a ton of information on it that is hard to discern when zoomed out. For that reason, before we jump into the different pathways, there are a few core principles of metabolism that we need to establish to make future concepts easier to understand. First, we need to define and understand the broad strokes of what metabolism is. To put it very simply, metabolism corresponds to the chemical processes used by the body to maintain life, and it typically can be separated into catabolism and anabolism. Chemical reactions under catabolism are associated with the degradation of nutrients to extract their smaller components or make energy. In opposition to that, anabolism corresponds to reactions that take smaller components and make larger biomolecules, typically for the purposes of storing energy. As a quick example in our model cell, when one consumes food that contains glucose, this glucose gets absorbed in cells and can either be degraded via glycolysis, the citric acid cycle and oxidative phosphorylation, to produce energy in the form of ATP, or it can be used to form a longer chain of glucose residues, called glycogen, to store that unneeded energy. Likewise for fats, for example fatty acids, catabolic reactions such as beta oxidation and subsequent pathways like the citric acid cycle and oxidative phosphorylation will degrade the long carbon chain to make energy. Anabolic reactions, on the other hand, will use the fatty acids to create larger molecules called triacylglycerols in order to store the fat in the body. Obviously, some of the pathways that I have listed here, such as glycolysis, the citric acid cycle, and so on, are pathways that we will cover later, so the details are not important right now. The takeaway message from this is that metabolism is really about equilibrating the catabolic and anabolic reactions to meet the needs of the cell. So, if the cell needs energy, it will use glucose and fatty acids to obtain it via catabolic reactions, and inversely, if it doesn't need it, the cell will store its nutrients with anabolic reactions. Now, in order to understand the general rules that guide these pathways, we need to discuss thermodynamics. Thermodynamics is a branch of science that deals heavily with energy, and as we've just seen with catabolism and anabolism, energy is a fundamental aspect of metabolism. As such, I want to review a few topics that will be helpful tools for us later. For the purposes of this video, what matters and what I will focus on is simply building an intuition on key principles rather than knowing how to directly apply any equations or whatnot. So, to get us started on that topic, I want to first consider a simple reversible reaction of reactants A and B yielding the products C and D. Generally speaking, what thermodynamics allow us to understand is whether the reaction from any reactants to products will happen by itself or, in other words, spontaneously. If not, then the reaction will require an input of energy to happen. Also, thermodynamics allows us to understand which reactions are the rate-limiting steps to pathways. So, the tool to quantify this spontaneity is Gibbs free energy, which we can obtain from this equation. Free energy G equals enthalpy H minus temperature T times entropy S. Since we are not going to discuss how to derive this equation because it goes a bit outside the scope of this video, I suggest consulting other resources if ever that aspect is important for your studies. Now, simply put, H can be interpreted as the total energy of the system, S as a measure of disorder, and G as the available energy of the system. So, when you subtract the product between the entropy and temperature, which increases this measure of disorder, to the total amount of energy, you obtain the free energy in that given system. This free energy is something that is both present in the products and reactants, and because of that, it is often more insightful to consider the change in free energy between the products and the reactants. This difference is denoted by the delta symbol that accompanies the free energy, enthalpy, and entropy of the system. Now, in terms of the equations, the value that is most important for us is the change in free energy, delta G, which will indicate the spontaneity of the reaction. If delta G for a given reaction is negative, then the reaction will be spontaneous and is said to be exergonic. On the other hand, a positive delta G implies that the reaction is endergonic, non-spontaneous, and requires the input of energy to occur. Besides the sign of delta G, if its value approaches zero, then the reaction is said to be at equilibrium, which simply means that it can go either way in the forward or reverse direction. Typically, in metabolic reactions, that means that high amounts of reactants will cause the reaction to go forward in order to equilibrate with products, and similarly, high amounts of products will equilibrate in the reverse direction with the reactants. Lastly on this, reactions with delta Gs that are very negative are said to be irreversible because the reactants will always make the products regardless of their initial concentrations. So, as you can see here, the direction of the reaction, and by association the free energy, can vary on some conditions such as the concentration of reactants and products. Given that, there is also a standardized version of the Gibbs free energy that is denoted delta G naught. This version of free energy follows the same rules for the signs of delta G, and it allows to give insight on the nature of the reaction. However, the true indication of spontaneity that happens in vivo comes from this delta G because it uses the physiological conditions of the reaction. Now, to exemplify these concepts of thermodynamics, I want to consider the values of delta G naught and delta G for the process of glycolysis. Although we have yet to cover glycolysis, the idea here is really to see how metabolic pathways are set up and talk about some of their governing principles, as a lot of these thermodynamic features will apply in other contexts. So, if we consider our table, the first column shows all the enzymes that make up the pathway. If we take a look at a few of the reactions of this pathway, the enzymes are labeled in light blue, so GAP dehydrogenase or GAPDH, phosphoglycerate kinase or PGK, and enolase. As we are on the topic of enzymes, there are a few aspects and properties about them that I want to introduce. The first important aspect about them is that they increase the reaction rate, or in other words, they basically make the reactions possible without interfering in them. Because of this, enzymes are also referred to as catalysts. One common way that enzymes manage to achieve this is by bringing the reactants close together in order to properly orientate them and facilitate the reaction. The second important aspect about enzymes is that they mediate their activity at physiological conditions, so conditions of the human body, and this is very important to consider given that typical chemical catalysts operate at extreme conditions of temperature, pressure, and so on. Thirdly, again, in comparison to chemical catalysts, which are typically non-specific, enzymes are mostly specific to a given set of substrates. For example, a reaction between A and B yielding C can be mediated by enzyme A, but the opposite reaction of C yielding A plus B can be mediated by enzyme B. So not only are the enzymes specific to some substrates, but also to a given reaction. And in the same train of ideas, the reaction of A plus D yielding E can be mediated by a totally different enzyme. This aspect of substrate specificity is especially important when we consider the fourth property of enzymes, which is the fact that they can be regulated. Given that the goal of metabolism is to balance out the anabolic and catabolic reactions, regulation of the enzymes that mediate the reactions is crucial. The three main mechanisms to do so is through allosteric, covalent, and gene expression control. If we first consider the allosteric control via the A plus B gives C reaction, allosteric control comes from the binding of substrates to the enzyme, which will either stimulate or inhibit its activity, here denoted by the green and red arrows respectively. Covalent control, if we consider our reaction again, arises via the interaction of a second enzyme that will covalently modify the structure of the first enzyme. Here again, this modification, which is typically a phosphorylation, will either stimulate or inhibit the activity of the first enzyme. Finally, gene expression happens at the level of the nucleus, and given that enzymes are simply proteins, the rate of expression in the cell can control their influence on metabolic pathways. With this aside in mind, we can now discuss the delta G values of the enzymes in this table. Here, the second column represents the delta G naughts at standard conditions, and the third column represents the physiological delta Gs. You will notice that in some instances, the delta G naught is positive, which would imply that the reaction is endergonic and would not happen, but at physiological conditions, especially with the aid of enzymes, all the reactions are exergonic. The fact that all the reactions end up being exergonic makes sense as we want the pathway to run forward. Another aspect you will notice is that there are two types of exergonic reactions, those with very large negative delta G values highlighted in yellow, and those with delta Gs close to zero highlighted in green. Recall that delta Gs close to zero are reversible reaction, and in the map, they are denoted by the double-sided arrows as you can see on the left in the zoomed-in pictures. The irreversible reactions, on the other hand, are illustrated as the one-sided violet arrows. If you recall what I just mentioned about the property of enzymes and the fact that they are regulated, you will notice that the enzymes for these reactions receive plenty of regulatory arrows, and that makes sense because they catalyze the most exergonic reaction of the pathway. As such, these reactions are also known as the rate-limiting steps. While covering the different pathways, you will notice that I will typically spend more time covering the rate-limiting steps due to the heavy control they tend to receive. Now, if we go back to our definition of metabolism to make sense of what we discussed on thermodynamics, as we said, catabolic reactions mostly serve to produce energy in the form of ATP. However, when it comes to anabolic reactions, it turns out that these reactions are mostly endergonic and non-spontaneous on their own, so for them, they will need an input of energy to make the reaction exergonic. Hence, you can see that the energy produced with catabolism can then be reinvested through anabolism, and this is the basis of our life cycle. We eat food to extract its energy, and we use that energy to grow and fuel the reactions that would otherwise not happen. Now, to understand how that happens, we need to briefly discuss what ATP actually is. Let's begin our discussion with a bit of terminology to understand the structure of adenosine triphosphate, or ATP. So, ATP is a rather simple molecule composed of a 5-carbon ribose and a purine base known as adenine. Altogether, the ribose and adenine make a molecule known as adenosine. Now, where ATP gets interesting is with its phosphate groups. Adenosine plus one phosphate group makes adenylate, or adenosine monophosphate, or simply AMP, and two phosphate groups make adenosine diphosphate, or ADP, and as you can expect, three phosphate groups finally makes ATP. With these phosphate groups, there are two important names of bonds. The bond between the adenosine group and the first phosphate group is called the phosphoester bond as shown in red, and the next two bonds between the remaining phosphate groups are called phosphoanhydride bonds that are shown in orange. Now, the reason why ATP is used as a source of energy can be seen when we consider a table of delta G naught values for the hydrolysis of ATP. As a quick note on hydrolysis and what it means, hydro refers to water and lysis refers to a cleavage or cut. So, in other words, the hydrolysis of ATP is the cleavage of its phosphoanhydride bond by water. The cleavage reaction causes ATP to lose a phosphate as an OH from water goes to the phosphate group, and by itself, a phosphate is typically denoted as Pi. So, to come back to the table, you can see that the hydrolysis of ATP to yield either one or two phosphate groups is very exergonic and releases a ton of energy. This energy that is released can then be harnessed by the process of reaction coupling, which is simply the process by which the hydrolysis of ATP can be coupled to an endergonic reaction to make them exergonic and thus happen. To exemplify this process, I want to consider a classic example of this coupling here shown on the left. Here again, this is a reaction from glycolysis and it actually corresponds to the first step where exokinase catalyzes the addition of a phosphate group onto glucose to make glucose 6-phosphate. If we examine this reaction in a little bit more detail, we can see that there are two main reactions at play. the addition of a phosphate on glucose to make glucose 6-phosphate, and the hydrolysis of ATP to ADP, which we know is a highly exergonic reaction. On the other hand, the addition of a phosphate group on glucose is endergonic and does not happen spontaneously based on the delta G naught. To salvage this, it turns out that the delta Gs of the two reactions can be added together, and the free energy released by the hydrolysis of ATP makes the overall reaction exergonic. Back to our metabolism definition, we can tweak our model slightly to mention that the energy used is ATP hydrolysis for the most part. Now, one thing about thermodynamics that I haven't explicitly mentioned, but is well illustrated with the hydrolysis of ATP, is that if a reaction is spontaneous or exergonic in one direction, then it will be endergonic or non-spontaneous in the opposite direction. Consequently, the opposite reaction of adding a phosphate group to ADP to reform ATP demands energy to do. As such, this is where the energy supplied by catabolic reactions, here in red, is important to allow the recycling of ADP and keep the metabolic cycle going on. Now, the last general topic that I want to cover is a particular type of reaction that comes up again and again in all metabolic pathways, and this reaction type in question are the oxidation-reduction or redox reactions. This type of reaction is ubiquitous all across chemistry, and I know that some teachers like to give the typical example of iron oxidation to explain this process, even when it's covered in the biology class. Instead of doing that, I want to consider some concrete examples of these reactions in metabolic pathways because most redox reactions in the biological setting involve the same recurrent substrates. So, the most common occurrence of redox reactions in metabolic scenarios happens with two compounds called NAD and NADP. In the metabolic map, reactions involving NAD are shown in blue, and reactions involving NADP are shown in green. Before we talk about what these two compounds do in the cell, I want to briefly introduce their structures. It turns out that NAD and NADP are quite similar to each other, with the main difference being located in one of the groups attached to the adenosine segment of the molecule. At the place of the red X, NAD simply has an hydrogen to make an OH group, whereas NADP has a phosphate group attached to the adenosine segment. If we go up on the molecule, for both NAD and NADP, there are two phosphate groups linked together to attach the adenosine to a ribose and eventually to a nicotinamide group, which is where both molecules operate their role. What you see right now represents the oxidized form of NAD and NADPH, and if the two molecules react with two hydrogens, the two molecules get reduced to NADH and NADPH respectively. If you are unfamiliar with NAD and NADP, you might be wondering what is the point of having the two molecules if both can perform the two reactions reversibly. The reason why both are crucial in the cell is because the NAD supply represents the oxidation power of the cell and NADP the reduction power. As a quick aside, in my personal experience, I struggled for the longest time with quickly grasping what a reduction or an oxidation was because the terminology was not intuitive to grasp for me. It always used to take me a minute to identify which reaction was which, and I'm sure that some of you might be in the same boat. What I eventually grasped over time is that as a whole, oxidized substrates become less complex and reduced substrates become more complex. For example, glucose, through the process of glycolysis, becomes less complex as it gets broken down to pyruvate, and this pathway represents an oxidation. Inversely, acetyl-CoA is a small molecule of a few bonds if we exclude the CoA that gets taken off, and it can get reduced to produce fatty acids, which are long chains of hydrocarbons. By the way, given the space constraint of the map, the R in the fatty acid is a shorthand for all the long bonds. This idea of complexity can even be seen in the oxidized and reduced forms of NAD and NADP. Indeed, although it is minimal, NADH and NADPH are more complex than NAD and NADP because they both have an extra hydrogen and thus make the reduced version more complex. Now, for something to gain complexity, or in other words, get reduced, something else must get oxidized or lose complexity. This is where NAD and NADP differ in function. If we first consider NAD and NADH, their ratios in the cell indicate that there is much more, about 1,000 times more NAD than NADH. This large amount of NAD can then get reduced to NADH in order to oxidize the substrates. So, as you can see in the example boxed on the left, malate gets oxidized to oxaloacetate as it loses that hydrogen to the newly formed NADH. On the other hand, NADPH outnumbers the amount of NADP by about 100. This high quantity of NADPH gets oxidized to NADP and in the process gives away its hydrogens to reduce substrates. The example in the green box on the left illustrates this as the oxygen gains in hydrogen given by NADPH. An alternative and more scientific way to talk about the complexity that I've kept mentioning is to say that oxidations are a loss in electrons and reductions are a gain in electrons. Since electrons are shared between bonds, an oxidation will typically involve losing a bond and a reduction will make substrates gain bonds. So, to reformulate this idea one last time, oxidations cause the loss of complexity or electrons of a substrate, and this is accompanied by the reduction of NAD to NADH. The NAD basically takes away the electrons to make NADH. Inversely, reductions are the gain of complexity or electrons, and this is achieved by the oxidation of NADPH, which gives its electrons to the substrate. I know that this terminology can take some time to set in and may be confusing at first, so do not hesitate to ask away in the comments if anything is unclear. With this being said, this concludes our small note on redox reactions, and we are ready to summarize the general metabolic concepts before we start discussing the different pathways. So, when we consider what metabolism is in essence, it is really the balancing out between anabolic and catabolic reactions. Catabolic reactions are overall very exergonic and serve to break down nutrients and possibly produce the energy to synthesize ATP. On the other hand, anabolic reactions are generally endergonic and require a source of energy such as ATP hydrolysis to fuel the reactions. Now, for both catabolism and anabolism, there exist a ton of different pathways that serve to degrade or build something from the nutrients we ingest. Regardless of the nutrient, all the different pathways share certain commonalities. First, a class of proteins called enzymes mediate the reactions of the pathways and typically fall into two categories of reactions: those that are reversible and depend primarily on the availability of the substrates, and those that are irreversible and constitute the rate-limiting steps of the pathways. More than often, the rate-limiting steps of metabolic pathways are the sites at which the enzymes mediating the reactions get regulated via allosteric, covalent, or transcriptional control. This control typically allows cells to selectively push nutrients towards a certain fate, either anabolism or catabolism. Another similarity across the pathways is the presence of redox reactions that either serve to oxidize substrates and degrade them, or reduce the substrates to build something. As such, substrates in anabolic reactions often get reduced via the oxidation of NADPH, and substrates in catabolic reactions get oxidized by the reduction of NAD+ to NADH. The last important detail to mention with respect to our metabolic map is that there are two major sites where metabolic reactions can happen that we will consider. First, there are the reactions that are compartmentalized to the cytosol, and second, those that are happening in the mitochondria. So, you will see throughout this discussion that we will mostly move around between these two areas. Alright, now with all these tools and concepts introduced, the metabolism of the first nutrient I want us to discuss is carbohydrates, and to get us started, I want to build a little overview to guide our discussion. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.